Okay, so here we would like to review from lectures 14 onwards, so 14, 15 and 16, where in the first half we would like to describe more about electro-osmotic flows and silicon microchannels, where we would take a surface to the point of zero charge, and then just above it or below it to have a dual layer of charge created, and then would study the aspect of flow or mobility associated with such a dual layer which has been created under the influence of a potential which has been provided. So here we would typically design a crisscross kind of electrokinetic network, where through Thevenin's network theorem we would like to as a function of no current being generated at a particular node, would like to see what is the charge movement, what is the mobility associated with the longest channel. So we complete this particular design and then go on to a very interesting area, which is the real important aspect of biomems, which is the introductory concepts into molecular biology, or the biology of DNA or other molecules as we know it better from a standpoint or from a perspective of diagnostics. Okay. So here we first reviewed the basic cell, and we tried to analyze the cell in terms of the various different organelles which are present in such a cell and various different small moieties, and this we did from a size aspect, particularly how we would arrive at the region which is central to the nucleus of a cell, which is also known as the chromatin region, where there would be the presence of the chromosomes, which are long, uninterrupted, packed, supercoiled, linear polymer strands of deoxyribonucleic acids. We discussed about the tentative sizes of human chromosomes to be about close to 50 to 400 into 10 to the power of 6 units long, and this typically would be a few centimeters. And we also described that Mother Nature is such a good assembler that it packs such a 5 or 6 centimeter long object into a very small volume, which is close to only a micron cube, which is the nucleus. Okay. So one of the greatest integration densities that can ever be achieved because of this folding nature provided in the very simple unit of cell, that is the nucleus of the cell. We started unfolding the chromosomes, and through TEM as well as physical cartoons arrived at the double stranded DNA structure, which is also one particular helix within this chromosome. Okay, and such many folds of such double strands would actually make what you call the chromosome. And then we looked at the DNA structure to be two different strands, with one strand composed of a sugar phosphate backbone comprising of a 5 carbon sugar and a phosphodiester linkage between two such sugar molecules, and also bases which would be 4 different bases in numbers: adenine, thymine, cytosine and guanine. We looked at the different bases in terms of its chemistry of bonding to each other. Normally this would be hydrogen bonded to each other, and what we also observed was that two moieties like cytosine and guanine would be bonded with 3 hydrogen bonds, and the other two, adenine and thymine, would be bonded with 2 hydrogen bonds, and there would be something associated with the stringency of the binding process, and the binding process we would also know as hybridization. So basically it was a phenomena associated with the opening of the or unzipping of this double stranded DNA because of the exposure to a certain high temperature, followed by a reversible association or linkage of this DNA, again under a certain critical temperature, where we could provide sufficient kinetic energy to bring them close enough to start rebonding again. Okay. So in one case, as a function of temperature, you are completely debonding the DNA. In another case, again as a function of temperature or kinetic energy, you are rebonding them, and this process or phenomena would also be known as hybridization. And again what we discussed is that hybridization could be a function of the of course the temperature, the salt and buffer concentrations which are present the GC content, the guanine cytosine content of a particular DNA, and so on and so forth. And this would typically happen between a DNA and a DNA or a RNA, and then again an RNA and RNA. So all this hybridization phenomena would be very important from a diagnostics perspective, as we will see later in one of the other lectures. We talked about two different cases. One is the reduced stringency case of hybridization, where there would be many mispairs or mispairings between the different groups on such a double stranded DNA. For example, if we were to bond a cytosine to a cytosine, or even a cytosine to an adenine, there would always be a misfit, because in one case there would be an additional hydrogen dangling bond which would be unsatisfied, okay, and in the other case there would be a complete disparity because of size issues, etc. And therefore, in such cases the bonding would typically not happen, okay, or when it happens very mildly. And on a statistically more significant basis, if such instances of reduced stringencies are available within a molecule, the molecule would split apart very easily, and it would not obey the particular melting temperature phenomena that would be associated with an otherwise normally available DNA. So we talked about the various theories of association related to these base pairs as related to the finally the composition of the whole DNA molecule. So we covered a very interesting phenomena called the polymerase chain reaction, which was invented by Mullis in towards the beginning of 1980s, and we also described about how an enzyme, that is the DNA polymerase, could be used for suturing the various base pairs which are commonly available as triphosphates within the PCR reaction, thus making copies of a template DNA which would initiate or which would start initially from the solution. We also investigated what is the use of such polymerase chain reaction towards diagnosis, forensics, so on and so forth, and then briefly talked about how the 3-step PCR process could be obtained on a laboratory scale by preparing different temperatures, either through a Peltier driven heater cooler assembly, or even through as basic components like differently heated solutions, into which you would immerse this reactant for many times, so that you could have exact copies of the molecular DNA, the template DNA. The more number of times you circle thermally across these 3 different temperatures, which are 95 degrees for the denaturation, the around 54 degrees for the annealing, and again 72 degrees for the extension, you would have many number of copies of the same DNA strand. And also we talked about the various ways of calculating the rates of such PCR reactions by looking at the speed at which the enzyme would suture the DNA to extend the DNA, particularly in the extension step, the last step, that is at 72 degrees Celsius, and which is incidentally also the rate limiting step of the whole process. And we found out that enzymes with all sort of speeds varying from 30 to 100 base pair per second suturement are possible or possibly available. And in a way, if you look at real time what is going on, the reaction rate limits itself because of the unavailability of the various other participating species in the solution of interest, etc. So in that sense, we derived the amplification efficiency of such a reaction, where we talk about a factor gamma defined by 1 plus efficiency PCR for a n cycle process to the power of n. Typically the efficiency PCR would be 1 for the first 20 cycles or so, but beyond that there would be a rapid deterioration of the efficiency of the PCR process owing to the fall down of concentrations of the various participating species. For example, the DNTPs or the dinucleotide triphosphates, or even the let us say the solutions which are containing magnesium ion, etc., they would deteriorate rapidly, and also the availability of the primers would also reduce quite a bit, because the primers also are finitely available within the solution, and therefore, there is a self plateauing action which would formulate. So, the first 7 cycles or probably first 10 cycles of a PCR process would actually result in a growth of PCR at a linear basis, and then it would suddenly have a 2 to the power n type of an exponential increase followed by this plateauing. The first 7 or 8 steps or cycles would typically not result in complete yield because of the fact that still there is a lot of diffusion limited reactants spread around as conglomerates and not properly mixed or uniformly mixed. Once the mixing is complete, then every PCR would result in exact 2 copies, okay, of whatever molecular concentrations are present till and until the concentrations again fall down. So, we described the efficiency of the PCR process in this particular manner, and then we also looked at the various determining steps of transduction where this PCR process could be gauged. Okay, and some of the steps would include things like or dyes like cybergreen or Tachman PCR or even molecular beacon assembly, where what is happening within a reaction can be on a real time manner gauged in terms of the concentration of the falling down species. So, we would start with something like a simple cybergreen reaction, or where there would be a dye which would get intercalated into the double stranded helical structure of DNA, and there would be a release of or sudden increase in the quantum yield of the particular dye. So, the idea is that wherever there was such a cage like structure being deciphered, there would be a intercalation and there would be an increase in the fluorescence intensity because of the increased quantum yield of the dye in reference to that cage like structure. There would be an obvious fallout of this process that any double stranded DNA available in the solution would yield light signal, which would talk highly about its non-specificity of a recognition process, but that is a very important way, because ultimately the statistically significant numbers would reflect only the copied strands and not the other strands. If you are talking about a 20 cycle process, so on and so forth, where there is the involvement of millions of molecules, okay, and trace concentrations of other species which are present may not be significantly contributing to the overall growth of the light signal, so on and so forth. In this slide, we also described another Tachman based PCR, where it would be involving a dye pair between a fluorophore of one kind with a certain excitation and emission frequency, which would then be associated with the dye of the second kind with its emission, with its excitation in the same range as emission of the previous dye, and its excitation in a completely its emission in a completely different range. So, in this slide we described the FRET process, the fluorescence resonance energy transfer process, where whenever in a primer these 2 species or these 2 molecules are put together and they are separated by a distance equal to a Forster's distance, which is a very small number, there would always be a coupling of energy between the first dye of the pair to the second, resulting in the emission of the first dye going down and the emission of the second dye going up. Okay. And typically, if you could identify such a pair of dye associated with each primer, each binding event of the primer specifically to a DNA molecule would cleave off one of the dyes to go into the solution, where it would result in exactly the opposite kind of signal, where the first of the pair would have a growth in the emission frequency, and the second of the pair, which is already stitched onto the DNA, would have a reduction in the emission frequency. So, such a turnover of the intensity at different frequencies of this dye pair would definitely comment about the way the PCR amplification process is happening, and this would be a very good real time gauge or process for determining how the DNA is being replicated within a solution. A similar arrangement was described in terms of molecular beacon assembly, where there would be a quencher and a dye again present in a primer as a hairpin loop DNA, and the idea is that the primer would align itself flat and bind to a end of a certain DNA strand. It would result in the separation between the absorber and the dye, and the dye's signal would grow because of this binding. However, such molecular beacon assemblies are only present for a very minuscule section of primers, and they cannot be used as a generic technology for the whole PCR process. So, in a nutshell, this is what we described about how to read the PCR. We talked about how a thermal cycle associated with the PCR process could result in an increase in light signal in a real time PCR reaction, where we described a strategene real time PCR device with multiple wells, about 96 wells, high throughput assembly, and there we also further described that how the readout would happen typically across the 72-degree step, at the end of the 72-degree step, which is also the extensional step in the whole PCR reaction, and conglomeration of such steps as a function of cycle could be plotted, which would yield in that linear exponential and plateauing regime of the PCR reaction, which we described earlier for the PCR process as such. Following this, we also described how to design a PCR reactor on a micro scale, and for this the very important, the most important part would be that how the temperature would vary with let us say a surface which is heated already to a certain temperature, and a very small film of the PCR reactant contained within a small chamber sitting over this surface. Okay. And in that light, what we try to look at is a very important electrical analogy associated with such a temperature system, where there would be a heat capacitance and there would be a heat resistance associated with such a circuit. And then what we try to determine is that how the the whole reactor can be made into the heat resistive and a heat capacitive control, finally resulting in a RC circuit equivalent of the heat flow. Here the way that the heat is flowing dq by dt would be treated as the sort of current flow, and the way that temperature would be varying as a function of distance, or the temperature gradient would be treated more as a potential associated with that heat flow or current flow. So, by doing that, we finally arrived at the time constant for the RC circuit, which would typically mean it is a point where 3 times the time constant would be the time needed for the temperature to go to a set point. For example, in one case, if the temperature needs to be of the particular PCR mass needs to be going to 95 degrees Celsius, that is the set value of the temperature, and we need to wait for at least 3 RC times, which is again a function of the geometry of the geometrical parameters related to the chamber in the micro scale, and this way we could be predictively able to determine what would be the time needed for the temperature to go up to that particular set value. In order to design a control system using a proportional integro differential controller, which would ultimately be needed for designing a micro PCR, you need to go through these process steps of the RC circuit analogy or method to hit upon the right times which would be needed for that micro scale PCR process to go on. And in that light, we designed some using some problems or some design parameters, some micro PCR chambers, where we could estimate the overall PCR cycle time to be much smaller in comparison to the time that it would otherwise take for a bulk PCR process. So, this in a nutshell completes or gives a complete review of the lectures 14, 15 and 16, and also probably some extent of 17. What I am going to do in the next review is to sort of complete the remaining lectures associated with this subject of biomems and microfluidics. Thank you.